When we look at the internal structure of the eye, it's best if we view a sagittal section, and the three different layers are called tunics. The outermost one is called the fibrous layer, and it's made up of the sclera and the cornea. The sclera is this outermost layer of the eye, and it's continuous with the cornea. So the part that we can see here is the cornea, followed, and it continues to become the rest of the outer tunic, which actually is continuous with the dura mater of the brain back here. The vascular layer is the middle layer of the brain, or of the, sorry, the eye, and it includes the choroid. The ciliary body contains the ciliary muscles, which control the lens and the iris. And the iris is specifically made of two smooth muscles that constrict and dilate the eye. So it would be this middle layer of the eye that you can see right here. The innermost layer, which is where the phototransduction occurs, where light is basically transduced into nerve energy. So the neural layer contains the retina, which has the pigmented layer, and the neural layer. This is where we find photoreceptors, bipolar cells, and what are called ganglion cells. And the yellow part of this diagram is where you see the retina. At the very back of the eye, this becomes the optic nerve. Our next slide is showing the choroid layer, and specifically within the choroid layer, this is the vascular layer because there's blood vessels. There is the iris, and the iris is made up of two different muscles. There's what's called the sphincter pupillae and the dilator pupillae. And both of these are smooth muscles. So that means they are under autonomic control. So they automatically respond to light. So the dilator pupillae muscle, when it contracts, it dilates the pupils. So this would be the sympathetic nervous system, which causes that to happen. And the sphincter pupillae, when it constricts, the size decreases from the parasympathetic nervous system. So on the right, we have the sympathetic nervous system shown up here, where the pupil dilates. And over here, we have the parasympathetic nervous system, where it constricts. On our next slide, we can see the microscopic anatomy of the retina. So this would be the interneural layer. And here we can see what's called the optic disc. And the optic disc is also called the blind spot. And that's because it lacks photoreceptors. So when the light touches that part of the retina, there's actually no image that's formed. So the retina has 250 million photoreceptors, and those photoreceptors are probably what you've heard about before, rods as well as cones. The rods would be for night vision, the cones would be for daytime or color vision. And within the cones, there is a high concentration of cones at what is called the macula lutea. And it's the very posterior pole of the retina that's lateral to the blind spot of the retina. At the very center of the macula is a very, very small, tiny dot, which we'll see better coming up. That's called the fovea centralis. So it's a pit in the center of the macula lutea that contains all cones. So it's the best visual activity. I like to think of that almost as like the best HDMI video that you could possibly have. So when we look at those three cells, we can see them shown here. The first cell would be the photoreceptor, the rod or the cone. So what happens? This is at the very back of the retina. So this is the pathway of the light. So the cornea would be in this direction here. So it's coming this way, the light as it enters through the cornea, through the aqueous humor, the lens, the vitreous humor, and then finally coming in contact with the retina. It first stimulates the photoreceptors. So the signal goes in the opposite direction. So the photoreceptors or the cones are what are called bleached. There's a chemical reaction that occurs in that. Then there's a neurotransmitter released from the photoreceptor to the bipolar cell. The bipolar cell then releases another neurotransmitter to the ganglion cell. And that's where the nerve impulse is generated. So the action potentials now are going to travel down the axons of the ganglion cell into the optic nerve. So let's look at the difference between rods and cones. Rods are for non-color vision. Cones are for color vision. And there's three visual pigments specifically for color vision. The rods function in a high sensitivity environment. So they function in dim light. So it's what allows you to see images at night. Whereas cones function in low sensitivity and function in bright light. The rods have low acuity, which means that many rods converge onto one ganglion cell. So they're not as precise. There's not going to be as precise a video signal essentially traveling to the optic nerve. Whereas the cones have high acuity because there's basically one cone per ganglion cell in the fovea, which remember the fovea is going to be that central pit in the very center of the macula luteate, which we'll see in much more detail in our next slide. The rods are much more numerous than the cones, and the rods work best in peripheral vision, whereas the cones are mostly in the central retina, which is in this macula luteate area. So the further we move away from that fovea centralis, the less cones we get, more rods we get. So when your eye doctor looks at the back of your eye with what's called an ophthalmoscope, this is what they see. So first we can see the optic disc, and remember that the optic disc is also called the blind spot. And it's called the blind spot because there are no photoreceptors. So this is why you need to turn your head to look into your blind spot before changing lanes. Remember what you learned about in driver's ed. So we're looking at the back of the retina. Now, just lateral to the optic disc is the macula luteate. This is the highest concentration of cones. And I like to think of it kind of like a dartboard. If we think of a dartboard, there's my poorly drawn circle. In the very center of the dartboard, where the bullseye is, that would be our fovea centralis. So that's the highest possible concentration of cones. Our next slide is showing what's called the aqueous humor. And the aqueous humor is fluid that's located between the cornea and the lens. The aqueous humor is formed by filtration from capillaries in the ciliary process. And the ciliary process is there's capillaries located there. Aqueous fluid then flows from the posterior chamber through the pupil. So the posterior chamber is located right back here in this area. That's where it flows into. And it flows then through the pupil to the anterior chamber. So the anterior segment actually contains aqueous humor. So humor means it's a liquid. Think of the liquid that's in a joint. We call them the humors. So the aqueous or the anterior segment contains the anterior chamber and the posterior chamber. So the lens and the ciliary zonule separate the eye into two segments. This is the ciliary zonule, also called the suspensory ligament. And this is the lens here. I'll highlight it for you so you can see it. That's going to separate the chambers into the posterior segment and the anterior segment. So the posterior segment is not the same thing as the posterior chamber. That doesn't seem like a big difference, but it is. So the anterior segment has two chambers, and the posterior segment is located posterior to the lens. So the aqueous humor is going to be removed from the eye. It's created with filtration, and it's replaced. It's a plasma-like fluid continuously formed by these capillaries. And those two chambers are shown here. But in the posterior segment, the vitreous humor is also liquid. It's like a liquid gel, and it's never replaced. So we're born with the same vitreous humor that we die with. This next slide shows a couple different homeostatic imbalances of the eye. This first image up here is glaucoma. And glaucoma is a condition in which the drainage of the aqueous humor is blocked. So fluid backs up and increases pressure within the eye. Pressure may increase to dangerous levels and even compress the retina and the optic nerve. So it could lead to seeing halos, blurred vision, and there's an increase in intraocular pressure. So there's different medications that relieve that pressure, one of which is marijuana. The next one is called a cataract, and this leads to a clouding of the lens. Sometimes it's congenital, and the lens can be replaced surgically with an artificial lens.